Hello everyone, this is Dr. Sang-Yong Lee. I'll be giving you a short introduction to one of our classes that are offered within the program. So the title of the course is BIOT110, Biopharmaceutical Product Manufacturing. This is a 3-credit class, which includes 2 hours of lecture and 2 hours of lab. But we offer this class in the spring semester only and within the 8 weeks format. So actually the contact time will be doubled because it's offered within the 8 weeks. And typically we offer this class as a hybrid format. The lecture will be offered virtually on the Canvas platform, but the lab will be face-to-face. You have to come in and do the hands-on lab on campus. Another important thing is this class is one of the 6 classes that are required for the Pharmaceutical Manufacturing Certificate class. And pharmaceutical manufacturing certificate was developed to meet the strong workforce demand within our area. So we have companies like Catalan and Baxter. They are doing the contract biopharma manufacturing service for the major pharma companies. So they are hiring a lot of workforce in this operation area. So there's a strong demand. So we developed this class to meet that demand. So especially we worked with some of our local subject matter experts. So actually I worked with 3 of the experts came from Catalan and Baxter about several years ago. Using a grant from the National Science Foundation, we developed the teaching resources such as textbook and then this course content. So it's a very valuable class for our program as well as our local employers. And if you talk about the drug product manufacturing process within the biopharma area, there's so-called drug substance manufacturing, where the active pharmaceutical ingredient is manufactured from the genetically modified cells, like Chinese hamster ovary cells. And once the molecule is manufactured, then you have to capture and purify using different purification methods like chromatography operations and filtration. And once that's done, and the drug will be formulated and aseptically filled within the primary containers. And then once that's done, the labeling and packaging operation will be done. Of course, there's a visual inspection of all the vials done. So we're talking about that process: formulation, fill finish, and inspection, labeling. So that is the main content of this class. Okay. So if I talk about the course content, of course, we introduce a lot of industry-specific terminology. For example, when you say parenteral drug, what does that mean? That's injectable drugs. So most of our biopharmaceutical drugs are injectable drugs. So besides that term, there are so many different words that are used by our industry. So we introduce those. And then the history of the parenteral drug development and the FDA guidelines or regulations on those drug manufacturing, we'll talk about those. So that's the basis of the CGMP and the requirements. We talk about those. And then in terms of a facility design and requirements, there's a strict guideline, of course, everybody has to follow. And then we'll introduce the barriers and isolators technology used for the manufacturing these days. And then, of course, contamination control methods of different kinds. Aseptic operation is one of them. We talk about those. And then different types of sterilization methods, including steam, light, and the filtration, gas. There are so many different types of sterilization methods. But, of course, for your biopharmaceutical products, you cannot use any other methods but the sterile filtration. So we'll talk about the importance of those. We also talk about the formulation principle. So once your drug is prepared, and your drug should be mixed up with excipients that can stabilize your drug and protect your drug during the shelf life. So we talk about the science of the formulation, what things are mixed and why they are needed in the formulation. Okay, and then once that's done, we talk about sterile filtration, which is considered as the final filtration required by the FDA. And then we talk about filling operation using different automatic filling machines, and then stoppering and sealing operation. We talk about those. So you can see syringes and cartridges, and then freeze-dried drug product right there. And you see the employers trained on the aseptic gowning, and then gowned up surfaces tested there in the picture. And then beyond those, we also talk about lyophilization, which is freeze drying, the science of freeze drying process, and then what things are required during that process. And we talk about visual inspection and the labeling operation, primary, secondary packaging operation, and then particular metal testing, and drug stability testing, because in order for you to decide the shelf life, you have to know how long your drug will be good. There are various types of testing methods out there. And then finally, we have a field trip at the end of the class, field trip to a local company like Catalan or Baxter. So students will have a chance to visit those companies and see how things are really manufactured. And then, of course, the course has the hands-on lab, which we do a lot of important hands-on skills during the lab process. So we do things like aseptic filtration and filter validation, destructible and non-destructible filter validation methods. And students go through aseptic counting process and environmental monitoring by using particulate counters, settle plates, and rotor plates. And then particle counters, viable counters, of course. And then aseptic drug filling and sealing, hands-on process. We do that. Students fill a small batch of vials with the media fill operation, so they can check the contamination possibilities after their filling. And then filled vials will be visually inspected. So we talk about the principle of visual inspection and what things will be considered as defects and what are the major defect categories and limit numbers, AQL limit numbers. And then we also do QC assays on drug products such as osmolarity measurements and coefficient water content measurements. So there are a lot of fun hands-on activities that we offer through this class. So I think this is it. Thank you, and I hope to see you in this class. Okay, bye.